Hello everyone, I am a Dr. Ruchira starting with another lecture of a microbiology that is bacteriology. Start with the gram positive cocci today. Gram positive cocci. It is of a divided at the time with the catalyst test. This flowchart is very important. Catalyst test is positive in the Staphylococcus aureus cocci, Staphylococcus aureus, and it is negative in the Streptococcus species. In the aureus, it is further divided on the basis of a coagulase test. In coagulase test, there is a slide or a tube testing. In slide, there is a bound coagulase, and in the tube, there is a extracellular free coagulase. Extracellular free coagulase is there. On the basis of a coagulase test, it further divided into pathogenic, that is positive test for the coagulase, and second one is the non-pathogenic, that is negative coagulase test. The example of a pathogenic is the Staphylococcus aureus, and the non-pathogenic is the cons species. Then it again divided on the basis of a novobiocin antibiotic sensitivity test. Novobiocin antibiotic sensitivity test, and this is performed on the MHA, that is Muller-Hinton agar. Muller-Hinton agar. In this test, there is a this is the culture media zone of inhibition. When there is a zone of inhibition, it is a resistance. It is a resistant one. And when there is a sensitive zone of inhibition is present. On this novobiocin antibiotic sensitivity, there is a sensitive or resistant bacilli are there, cocci are there. Sensitive is the Staphylococcus epidermis, and resistant is the Staphylococcus saprophytes. I am going to show this in my notes also. Screenshot this for better understanding and for scoring purpose. This is the picture of a catalyst test. In this we have see a bubble, that is a nascent oxygen bubble is there. In this test, there is a culture colonies added a 3% H2O2 in it, and cause this bubble like a nascent oxygen, and this is the positive catalyst test, and this is the negative catalyst test. Slide coagulase test looked like this. The positive side coagulase test having a clumps of bubbles, that is a pathogenic staphylococci, and in negative one there is no clumps present. This is the antibiotic sensitivity testing, that is novobiocin test, starting with a Staphylococcus aureus. Staph aureus. It is a gram positive cocci. It is having a grape-like structure, grape-like clusters. It is a catalase positive, and coagulase positive. The best selective media For Staphylococcus aureus is the MSA, that is mannitol salt agar. MSA is a best media. This is the MCQ type of question, which is the among the following, which is the best selective medium for the Staphylococcus aureus. So best selective medium is MSA, that is mannitol salt agar. And for this test, for this medium, there is a lumens medium. Then main virulent factor or antigenic factor. When main virulent factor or antigenic determinant for this is the teichoic acid. Again, this is another MCQ question. Which of the following is the main virulent factor or a antigenic factor for the Staph aureus? Is the teichoic acid, and second is the protein A. Protein A. Then clinically, the Staphylococcus aureus causes the food poisoning. Sometimes examiner ask about the uh, clinical question about the Staphylococcus aureus, that food poisoning due to the milk and milk product consumption like paneer causes by which of the following organism? Then answer should be Staphylococcus aureus organism. Food poisoning due to milk and milk product like paneer. Second, clinically it causes the SSSS, that is Staphylococcal skin scalate syndrome. Staphylococcal skin scalate syndrome. Scald syndrome. It is a exfoliative toxin A and B. In the children, causes the Ritter's disease. Causes the Ritter's disease in children, and in adult, it causes the toxic epidermolytic necrolysis. Toxic epidermolytic necrolysis, also called as a 10. 10 is causes in the adult, and in the child there is a Ritter syndrome. Toxic shock syndrome. Third one is the toxic shock syndrome. Also, there is a vaginal tampons during the menses, but this is not that much common. In the lab, there is a we have to take a sample of this patient, like pus sample, sputum sample, blood, urine, stool sample, on the basis of the Staphylococcus aureus grading. Then, in microscopically, there is a presence of a gram-positive violet color clusters. Uh, in this, there is a presence of a toxins, that is hemolysins alpha and beta are present. Beta shows the hot and cold phenomenon, and alpha shows the paradoxical actions. So, in this, also important is the in best selective medium, that is MSA, the colonies are uh, changed color from pink to yellow or golden color. So, this is all about the Staph aureus. This is the grapes clusters, like clusters, gram positive cocci, catalase positive cocci, coagulase positive cocci. Best selective media is MSA, that is mannitol salt agar. In this, convert the color pink to the color yellow or golden color. Then main virulent factor is the teichoic acid and the protein A. There is a presence of a two toxins, alpha toxin and beta toxin. And the clinically it causes the three diseases, that is food poisoning due to milk and milk product. Example is paneer, mostly from paneer. Then second one is SSSS, that is staphylococcal skin scald syndrome. in children, they causes the Ritter syndrome, and in adult, TEN syndrome. Then third one is the toxic shock syndrome. This is all about the Staphylococcus aureus. We have to move further on the other gram positive cocci, streptococci. On the basis of a blood agar, it is divided into three hemolyses: alpha, beta, and gamma hemolysis. Alpha, beta, and gamma hemolysis. Alpha hemolysis is the partial hemolysis. Beta hemolysis is the complete hemolysis. Beta hemolysis is the complete hemolysis, and gamma means no hemolysis or false hemolysis. There is a no hemolysis means gamma hemolysis on the basis of the blood agar. The alpha hemolysis having two examples. First one is the Streptococcus pneumoniae. Streptococcus pneumoniae. And second is the Streptococcus viridans. Streptococcus viridans. The beta hemolysis again divided on the basis of carbohydrate C protein. This is another MCQ. On the basis of a carbohydrate C protein, that is a lancified classification. Lancified classification is there. Which classification? Lancified. This is again a another type of a MCQ asked on the basis of a carbohydrate C protein. Which classification is there for this streptococcal? The answer should be lancified. Then in lancified there is a to V except the I and J type are present. Okay, A to V types are present except I and J. Then on the antigenic determinant M protein, antigenic determinant M protein, there is another classification called as a Griffith classification. Here is another MCQ on on the basis of a antigenic determinant, that is M protein, which classification is there? Griffith classification. This is another type of MCQ. In Griffith there is a group present: group A, group B, and group D. Group A means the Streptococcus pyogenes. Pyogenes is the group A. In group B there is a Streptococcus agalactin. Agalactin. And group D is the Enterococci. Enterococci. So this is all about the hemolysis of the streptococci. Further move about the types of a streptococci. We have to make a table for the types of a streptococci. The first type of streptococci is the streptococci pyogenes. Streptococci pyogenes. Second is the streptococci agalactin. Streptococci. Then third is the streptococci pneumoniae. Pneumoniae. And fourth is the streptococci viridans. Viridan. This is a very